Hello, everyone. Good afternoon. So Professor Goza says hi to you all, and she really wanted to be here today, but she couldn't because of her recovery from this medical procedure she went through. And my name is Maram Ganaiem. I'm here PhD student, and I'm gonna talk today about my project related to ADNP and autism. So we're gonna today together try to decipher the developmental aging mechanisms in cell culture. Especially, we're gonna talk more about the ADNP aberrant localization in the cytoplasm and in the nucleus. So what is ADNP? Activity-dependent neuroprotective protein, or in short, ADNP, is vital for brain formation and cognitive function. A complete knockout of ADNP leads to lethality in mice. No ADNP, no brain, and lethality. So we're talking about a very important protein. It has a very important role in the nucleus of the cell. It interacts with a lot of different proteins, and it is essential for expression and expression of proteins, and it also has an important role in the cytoplasm, where it enhances or stabilizes the microtubule and tau EB's interaction and stabilize the microtubules. So it has a very important protein also in the nucleus and in the cytoplasm. So while complete knockout is lethal in mice, if we look at mutations or de novo mutations in ADNP, we can see that they lead to a syndrome, which is called ADNP syndrome, and it's a developmental disorder, and the kid shows very characteristic features of autism. Not only in the developmental aspect, mutations in ADNP, but also somatic mutations in ADNP are found in the Alzheimer's disease brain. So here we can see the schematic representation of ADNP, and the arrows points for all the mutations or most of the mutations that are shown. In this presentation, I'm going to focus on two mutations. I will shed the light on them more in the talk, later on in the talk. And one important peptide that I'm going to talk about is here, small 8 amino acids called NAP. Just remember it for later on in the talk. Another related background, actually, Gozes and Chasman, who's sitting here, found that ADNP is part of the apoptotic pathway in neuronal cells. But more related background to my talk is that in the different other labs shows that different sites among the ADNP genes shows different signatures or episignatures, meaning they have different DNA methylations. So this will lead us to one of our scientific questions for today, which is whether those differences in DNA methylations leads to different phenotypes or distinct phenotypes in cell culture. This is one question. And another question, whether those we saw that ADNP has to do with mutations and development, and also in Alzheimer's and aging. So the question whether are major proteins involved in development and healthy aging related to our ADNP? To answer our first question, so you've heard a lot about SHANK3 in this session. So we looked for an interaction between ADNP and SHANK3. Actually, this work was done also together with Professor Boaz Barak and our lab. And what we found here was, what we tried to do, actually, we subjected hippocampal extract from ICR mice, as you can see here, to co-IP assays using actin antibodies in the presence of NAP, the small 8 amino acids that I've showed you, and skip other treatment. And we similarly probed here, as you can see, extracts from the mutations, the mouse mutations that Boaz Barak uses to test for SHANK3 tau ADNP interactions with actin. And what we discovered here was an interaction between SHUNK3 and ADNP throughout an SH3 domain that is among those two proteins. So if we look here at, I show you that ADNP has to do a lot in the cytoplasm. It's a very important protein in the cytoplasm, stabilizing microtubules together. We add to this picture now our SHUNK3. Another discovery that we found in our lab is the interaction between ADNP and other, and SIRT1, which is important in healthy aging. So ADNP is in both, interacts with SHUNK3 and now with SIRT1, which is important for aging. So here is the schematic representation of ADNP, and this is SIRT1, and we found that they interact throughout the CYP motif here, as you can see also in the silica. And we did that also in immunofluorescent and iPS cells, and with co-IP also. So answering our first question, ADNP is interacting with SIRT1 and SHUNK3. And now moving to the second and actually main question to the talk, which is, do mutations in ADNP lead to distinct impairments? To answer this question, we developed a new cellular system using CRISPR-Cas DNA editing system. We've created two different mutations in ADNP, one in the tyrosine 718 mutation, a stop codon mutation, and another one in the 403 mutation. We added GFP, fluorescent GFP, to the N-terminus of this gene. And we validated this model throughout immunoblotting. Actually, those two mutations, if you remember, they are in different sites among the ADNP genes, so they show different episignatures or have different DNA methylations. Our novel cell line that we developed in the lab shows very distinct phenotypes. First, increased neurite numbers in the proline 403 mutation, and a significant cell death in the tyrosine 718 mutation, in the proline 403, and a significant cell death in the tyrosine 718 mutation, as you can see here. So these results indicate for a differential effect of selected ADNP mutations. Each mutation shows different phenotypes. Not always. We also found that not only the proline 403 mutation has higher neurites, but they also have longer neurites. The neurites are longer, and as you can see here, we did immunofluorescence staining with tubulin and DAPI. That DAPI is not shown here. But what we found was that two mutations, now it's not working, but as you can see in the graphs there, ADNP and tubulin, both mutations show more localization in the cytoplasm than in the nucleus. Another exciting thing that we saw in these pictures was some cells in the tyrosine 718 mutation, we found some difficulties looking at the nuclear and cytoplasmic boundaries in the tyrosine 718. So we tried to look at this closely in live cell imaging, which is more sensitive to see these things. And what we did here, so you remember this green is showing ADNP, the GFP, and here are the two mutations. What we did here was, we did several treatments, control, no differentiation for the cells, and DMSO to encourage them to differentiate into neurons. NAP, if you remember our drug treatment, drug candidate, the 8 amino acid within the ADNP gene, and together with differentiation. And we looked for the localization of ADNP. We tried to see where is ADNP localized. And if you can see here, in the control, it packed nicely, the ADNP packed nicely in the nucleus, but in the tyrosine mutation, in both mutations, but it can be it looks more severe in the tyrosine, it is all over the place. It's fuzzy and in all different compartments of the cells. And when we treat those cells with NAP, we can see a very nice correction here in the graph, here in the figure and in the graph. So this was very nice results. And now we are asking, what is the mechanism in which NAP do so? How does NAP work to correct this localization? So what we did was adding NAP into confocal microscopy to see where it goes, first of all. And we found a new discovery that we didn't know before about NAP, in which it enters the nucleus of the cell, as you can see here. And it found in the nucleus, but less in the tyrosine mutation, and even around the nucleus. Not only in the nucleus, but around also. And we found also that this correction that I talked to you about in the first slide, it was 7 days treatment. Here, it was an immediate treatment. We can see this correction even immediately by adding NAP. Not like the same significant result as we saw after 7 days, but we did see some correction here immediately. Talking about the mechanism in which NAP corrects this aberrant localization. So we know from literature and before, from our lab, that NAP works on tubulin, on microtubules. It stabilizes microtubules, and it has to do with tubulin content. So we looked at the tubulin content in our clones, in the full-length ADNP and 2 mutated forms. And what we found here was a complete reduction of tubulin content in both mutations comparing to full-length ADNP. And we see that also by immunoblotting. So we thought that NAP works throughout stabilizing microtubules and corrects the localization of ADNP. So what we did, we took full-length ADNP with no mutation, and we treated it with zinc, which is known to disrupt microtubules' function. And we saw in this full-length ADNP, no mutations, just treated with zinc, same phenotype as we saw, this fuzziness of ADNP. And when we treat them with NAP, we see the correction again. And to validate this even more, we use nucadazole and paclitaxel, which one of them disrupt microtubules, and one of them stabilize even more. And we see the same correction after adding NAP. To conclude what I just showed you here, we show that ADNP interacts also with SHANK3, which is important in development, and with SIRT1, that is crucial for healthy aging, and that there is distinct cellular phenotypes that characterize different ADNP mutations, depending on the site of the mutation. And NAP, our drug candidate, rapidly enters the cell cytoplasm and nucleus and immediately corrects this aberrant distribution and throughout microtubule protection. And that's it. Thank you very much.